The exhilarating cycle of mitosis and meiosis, featuring Marc Teitelbaum and Shannon Zoldy. The cell cycle is an ordered sequence of events in the life of a cell, from its origin in the division of a parent cell until its own division into two. The cell undergoes a division of about 24 hours. Interphase is the first component of the cycle, which accounts for about 90% of it. During interphase, a cell that is about to divide grows and copies its chromosomes in preparation for cell division. Interphase consists of G1, the first gap, which lasts for 5 to 6 hours. S phase, DNA synthesis, the cell continues to grow as it copies its chromosome. This lasts for 10 to 12 hours. And G2, the second gap, it lasts for 4 to 6 hours as the cell continues to grow. Next is the mitotic phase, or M phase, which includes cytokinesis and mitosis. In mitosis, the cell divides in this phase. Next, cytokinesis is the division of the cytoplasm into two daughter cells. This lasts for less than an hour. Mitosis has two primary functions: growth and repair of cells, and the process by which organisms reproduce asexually. Interphase. The nucleolus and the nuclear envelope are intact. The cell copies its chromosomes in preparation for cell division. Prophase. The chromosomes are condensed. The nuclear envelope begins to fragment. The mitotic spindle begins to form. Centrosomes move away from each other, propelled by the lengthening microtubules between them. Metaphase. The centrosomes are now at the opposite poles of the cell. Chromosome centromeres lie at the metaphase plate. Anaphase is the shorter stage of mitosis. Chromosomes begin moving toward opposite ends of the cell. Telophase. The two daughter nuclei form in cell. Nuclear envelopes arise from fragments of parent cells. Cytokinesis is the division of the cytoplasm into two daughter cells. The results of mitosis. The daughter cells inherit the same number of chromosomes as the mother cell. Mitosis produces two diploid cells, each containing 46 chromosomes. The cells produced by mitosis are genetically identical to the mother cell and to each other.
Meiosis results in genetic variation. It allows for genetic coupling of non-identical DNA, resulting in an offspring which has the genetic material of two separate individuals. Diversity benefits entire populations. In meiosis 1, homologous chromosomes separate. In prophase 1, crossing over occurs. Chromosomes condense and spindle formation and nuclear envelope breakdown occur. In metaphase 1, pairs of homologous chromosomes are arranged at the metaphase plate. In anaphase 1, breakdown of proteins allow homologs to separate and homologs begin to move towards opposite ends of the cell. In telophase 1, each cell now has a complete set of haploid chromosomes. Cytokinesis occurs, forming two haploid daughter cells. There is no duplication of chromosomes. In meiosis 2, sister chromatids separate. In prophase 2, chromosomes condense again and the spindle apparatus forms. In metaphase 2, non-identical sister chromatids are positioned at the metaphase plate. Connected cores are attached to microtubules which extend from opposite poles. In anaphase 2, the breakdown of proteins allows sister chromatids to separate and move towards opposite poles. In telophase 2, nuclei form and chromosomes decondense. Cytokinesis occurs, resulting in 4 genetically distinct daughter cells. Results of meiosis. Recombination and independent assortment allow for genetic variation. Meiosis produces 4 daughter cells, each with a haploid set of 23 chromosomes. Mitosis versus meiosis. Meiosis reduces the number of chromosome sets from diploid, or 2N, which is 46 chromosomes, to haploid, or N, which is 23 chromosomes. Meiosis produces cells that differ genetically from their parent cell and from each other. On the other hand, mitosis results in diploid cells containing 46 chromosomes rather than 23. Unlike meiosis, mitosis produces daughter cells that are genetically identical to their parent cell and to each other.